Hi, everybody. Good. Okay, it looks like you can hear me. So thank you so much for coming to the second seminar of this semester. It is my pleasure to introduce our speaker today, Dr. Chris Martine. This is a rescheduled talk. Dr. Martine was the EEPB student invited speaker in the spring, and his original lecture coincided with the world ending, so we are really looking forward to still hearing from him today. Dr. Martine is the endowed David Burpee Chair in Plant Genetics and Research, the Director of the Manning Herbarium, and the Biology Associate Department Chair at Bucknell University, a primarily undergraduate institution in Lewisburg, Pennsylvania. He received his bachelor's in natural resource management from Rutgers before working for various state agencies as an environmental and outdoor educator. From there, he completed his master's in evolution and ecology from Rutgers and a PhD in botany from the University of Connecticut. Dr. Martine started his career as a professor at SUNY Plattsburgh for 6 years before moving to Bucknell, where he's been for the last 8. Much of the Martine Lab research features species of Selenium from the Australian outback, which we'll hear about today, investigating plant reproductive evolution and ecology, molecular phylogenetics, and new species discovery. Dr. Martine is a science communicator extraordinaire and the host of his very own YouTube series called Plants Are Cool Too, which everyone should check out immediately after this. And he was awarded the American Society of Plant Taxonomists Peter Raven Award for his exceptional outreach efforts to non-scientists. Lastly, speaking from experience as a former member of the Martine Lab, Dr. Martine is a wonderful mentor and has made undergraduate education and research a hallmark of his career. So with that, I'd like to pass it off to Dr. Martine.
Thank you so much, Emma. I appreciate it. I'm going to do what we usually do these days, which is the $1 million question, which is, can you see my screen? Yep. All right. So just give me a nod. Okay, great. So we're looking at some slides. Awesome. Thank you for that introduction. And holy cow, I just want to say, Emma, that we're also proud of you here and also impressed with everything you're doing out in St. Louis. So I appreciate the invitation from you and all the students, and I'm so glad you gave me this 2.5-hour speaking slot today. So see, this is the kind of thing where I'm going to imagine that everybody's laughing at my jokes because Zoom is crazy and I can't see anyone's reaction at all. So I'm just going to imagine you're nodding and applauding throughout this entire talk, which isn't actually 2 hours long. So as Emma mentioned, I'm going to be talking mostly about these plants in the genus Selenium, these things called bush tomatoes. And I'm going to sort of talk about it a lot as sort of like a lab-wide thing that we've been doing for a lot of years, right? And so we do a lot of different things in the Martine Lab. We think about where plants came from, how they got to be the way they are. We think about how they make more of themselves, how they interact with other organisms. As you'll hear about later on, we also get lucky every once in a while and get to describe new species. And across all of this, I'm also, as Emma mentioned, pretty focused on science communications. And I really like us to think about how we can sort of broaden the message surrounding our science and let more people know about what we are doing. And there's really two main threads that flow through all of this, right? One is that this is mostly about plants, and plants are pretty cool. And the other thing that it's mostly, all these things are involving students. And at Bucknell, we are a primarily undergraduate institution, also, as Emma mentioned. And so much of this work that you're going to hear about today includes students that are working either in our greenhouses, in our lab spaces, or whenever possible, out in the field with real organisms in real places. So I'm going to use words like we and us and our a lot in this talk. And what I mean really is all the people and mostly students that have participated in this research over the last few years. You can see these are the group that have sort of been in the lab in the last calendar year or so. Some of them graduating recently and some of them still working in the lab today. But I also need to note that throughout this talk, I'm going to be talking about work that has also had major contributions from some of the postdocs in the lab. Most recently, Dr. Angela McDonald, who's now at Chicago Botanic Garden, who was in the lab until last fall, and now Tanisha Williams, who's been in the lab since that time. And also need to also mention that Jason Cantley and Ingrid Jordan Thaden were previous postdocs in the lab, and I've been so fortunate to have these people here at a PUI as collaborators and co-mentors of our students. We're doing a ton of stuff in the lab. We have a lot of ongoing projects. We've been very lucky to get sort of state funding to do a bunch of PopGen projects on species of conservation concern here in Pennsylvania. We are extending some of those PopGen protocols to questions about biogeography across a number of genera in Northern Australia. And we've also had a front row seat as a lab group recently to see the incredible work that Tanisha Williams and her colleagues have done with Black Botanist Week. And that's been such an amazing thing to see happen and develop and to watch. And I do want to acknowledge sort of at the outset of this talk that I know that this is a really heavy day for a lot of folks. And so I'm hoping that, for the next 45 minutes, maybe we can just get sort of lost and transported into a world where we're just thinking about like flowers and plant evolution. But I do want to say to Tanisha and everybody that's been like so critical to these Black and STEM movements that we've seen this summer is that you're really making a real difference. And for 2020, for all of its warts, is going to be one of these years we look back on and say that was the start of something, right? And so appreciate the work that you've done in that regard. But what I'm really going to talk mostly about today are Australia's bush tomatoes. And so to start that, let's look at a flower. Let's get lost in this beautiful Australian Selenium flower. Look at that purple corolla. That's 5 petals fused together into a lovely stellate corolla. And then let's look at that wonderful yellow cone of anthers, right? And you may note that only the tips of those anthers are open and available for sort of like dehiscing pollen. And then check out that awesome sigmoid style emerging from the ovary that's clustered in among those anthers. And then that terminal nice green receptive stigma. And, I could just end here and we could just stare at this flower for a while. It's awfully beautiful. And it's part of the reason why I became so interested in this group. But another reason, and I won't just have us stare at that all day, is that, these plants live in a pretty neat place. And when I was given the opportunity to start working on these plants as an early PhD student, I can't lie, going to Australia was a big part of the decision to say, yeah, I'm totally going to do this work, right? And so what I'm going to tell you about today is really one lineage of Australian members of the genus Selenium. That's a big genus, right? It's a lot of species, maybe 1,400 species worldwide. And our work mostly focuses on something like 3 dozen, maybe 40 species of things we often call spiny Seleniums. They're loaded with prickles. They have other characters like stellate hairs that are pretty good synapomorphies for the group. And we're mostly focused on one group that's sometimes called the Selenium dioicum group, or more broadly, even the Selenium dioicum plus echinotum group, which really doesn't matter much to you, except for that's what we tend to call it. And these are taxa that mostly live across the northern sort of edge of the Australian continent, a place we call the Australian monsoon tropics that I'll probably just call the AMT from here on. And are mostly living in 2 regions you see identified on that map. In the west, the Kimberley Plateau, which is just an amazing place you should all go to. And then there in the east, the top end, which is sort of a nickname for the upper part of the Northern Territory that includes an amazingly cool place called Kakadu National Park, which is a name that'll become important later on. Most of these species are associated with sandstone outcrops and other sort of rock outcrops. So when we go there and we get out into these really remote locations, if we can find the rocks, we can often find our species. But the key reason that I got involved in this group and why I was so interested in them when this was sort of presented to me by my advisor, Greg Anderson, as a possibility at University of Connecticut, was that across this range of 40 species, what we see is some really interesting reproductive biology. In fact, across all these taxa that are more or less sympatric, all occurring across the same broad range, we actually see 3 different sexual systems, and 2 of them that are actually quite interesting, right? So what I'm going to talk about in a minute is sort of the hermaphrodite sexual system, which we consider to be ancestral in the genus Selenium, and then 2 really interesting sexual forms, one called Andromonisi and one called functional Dioecious. So let's start with the sort of easy one, right? Most of the angiosperms are doing this, right? It's a hermaphroditic condition where every single flower can do both male and female function. In this group that we are studying, there's somewhere around a dozen species that are doing this. Up until recently, they were sort of of an unresolved relationship to the other taxa that I'm going to tell you about next, mostly consisting of a big species complex that is not toxic, but taxonomically complex, right? Which might feel toxic sometime. And these flowers do what you think they can do, right? You've got that androecium consisting of 5 stamens, and those stamens make a pollen grain that has these 3 openings on it. Any one of those can have a pollen tube emerge from it and then deliver 2 sperm cells, thus completing male sexual function. These flowers also can do the female stuff, right? They have a gynoecium with an ovary and a style and a stigma. And if pollinated and then fertilized, these flowers can then form, those ovaries can become fruits that have seeds in them that become the next generation, thus doing female sexual function, right? So this is what most angiosperms are doing. This is what about 1/3 of our taxa are doing. There's about 14 species in our group that are doing that thing called Andromonisi, all right? And this is a really neat sexual system, right? The way this works is that every single plant in each of these species has a repeated inflorescence sort of structure, right? Every single flower cluster, every inflorescence consists of a hermaphrodite flower down at the base of the inflorescence, and then a number of male flowers that essentially subtend it, right? So we've got a bunch of those males at the top and a fully bisexual flower down at the bottom. And you might have 7 or 8 males or even 30 or 40 male flowers in sort of that more distal position relative to this hermaphrodite flower. It's a pretty neat system, right? And we see about 14 species that do this. But if you think Andromonisi is cool, wait a second. And these flowers do what you think they would do too. All of these males make functional pollen with the openings, and so does the bisexual flower, making functional pollen with these openings. So male function happens in both flowers, and then only that basal hermaphrodite is able to make a fruit and seeds. So there's your female function occurring in that flower, right? But the coolest thing, I think, among these 3 is something called functional dioecious, right? Now, dioecious may be a term you're familiar with. This is when you have plants that are separate male plants and separate female plants. Cannabis and willows and hollies are great examples. And if you were to encounter male and female plants in those taxa, it would be really obvious who's a male and who's a female, but that's not the case in these Seleniums. In fact, the only way that dioecious shows up in this genus, both in Australia and in 5 or 6 other sort of taxa around the world, is in this sort of cryptic hidden condition that I'll describe to you now. So in functional dioecious, in this genus, what we see are male plants, right, that make flowers that look male. If we look at this upper photo, what we see is a cluster of anthers, and those are making functional pollen, right? So these are male plants doing male function. What we also see in the same population of these species are plants that have flowers like you see on the bottom photo. And by eye, looking at that, you might say, well, that's a bisexual flower. That looks just like the hermaphrodite you just showed us when you talked about Andromonisi. And you'd be right. Morphologically, that's what's going on. And yeah, those flowers can do female function. That's a fully functional gynoecium. We have the formation of fruits. They're full of seeds. And yeah, those anthers make pollen. But that pollen looks quite different from all of the pollen images I've shown thus far, right? There are no openings. So a pollen tube cannot emerge and sperm cells cannot be delivered. So that pollen is non-functional. It's something we call inaperturate pollen. And the presence of that pollen in these flowers on plants that morphologically appear to be bisexual renders them only female in the functional sense. So we have our male plants that look like this and our females that more or less look bisexual, right? And this is absolutely fascinating stuff. And I was absolutely hooked when I read about this. And the first time I read about it was the beginning of my PhD, when I read this paper by Greg Anderson, my PhD advisor, and David Simon, who wrote the monograph on Selenium in Australia in 1980. And they were the ones who figured out that this is what was going on. They looked at the pollen, they tried a zillion crosses and found out that that pollen essentially never works. And that's why those plants are really functionally considered female plants. In writing that paper, they also hypothesized a way that this unusual form of dioecious may have arisen in the group, right? And their hypothesis was that on this sort of background of hermaphroditism that's ancestral in the genus, we first have this move to Andromonisi in the Australian taxa, and then that becomes the transitional system that gets you to this sort of dead end of functional dioecious. And it would happen basically in like three steps, right? First, you need some plants to end up with only the male cluster of flowers. And then now this plant is male. Now this plant makes functional pollen, and that's all it does. And then you'd need some individuals to basically end up with only this basal hermaphrodite flower, and then an additional step that would close up those pores in the pollen grain. And this is just sort of the leading hypothesis for how breeding systems and sexual systems would have evolved in this group since 1989, right? Now that's interesting, and we're going to come back to this because I'm going to talk about some of our recent phylogenetic work to sort of look about whether or not we have support for this. But if you're, learning about this for the first time, you might be thinking, okay, that's pretty interesting, but like, why, right? Why? I'm going to personify these plants for a minute and just say, like, if you're going to commit to being just male and just female, if you're going to commit to being obligately outcrossing, right? Once you are unisexual, there's no way to self-fertilize. So this is a big deal to do this, right? So if you're going to do that, let's say you're going to become female, right? Why would you bother with the energy expenditure required to not only make anthers, but fill them with non-functional pollen? Why in the world would you ever do that? Why would that happen, adaptively? And many of you are already probably reaching the correct conclusion in that it has everything to do with pollination. These flowers do not produce a nectar reward. The only thing that insects are doing when they visit these flowers is foraging for pollen, right? And you may know that in Selenium, this is a very tight association with really a specific suite of bees that do something called buzz pollination. They land on the flower, they clasp the tip of that anther cone, right? And there again, only the pores open for pollen. Their wings buzz is at a very particular frequency that then allows for the release of pollen out of those anthers onto their bodies, right? So this is a super tight association. So what that means is you really can't afford to get rid of the anthers because they're the thing that's bringing in your pollinators. In fact, the contrast of the anthers against the corolla is the big deal here, right? And bees are pretty smart, right? So they may learn that visiting a certain type of flower and not getting a reward is something they want to avoid. And so essentially you've got a really complex plant-animal interaction at play that leads to a pretty complex way of being obligately outcrossing as a dioecious species and a dioecious lineage. This is the only way, again, that Dioecia occurs in Selenium, and for whatever reason, there is way more of it in Australia than anywhere else on the planet. So this is the thing that I'm like super excited about. It's something that I've been studying for 20 years since I started my PhD, but really observing in a major way for the last 15. I've been really lucky for the last 15 years to go on 6 different expeditions to Northern Australia and do collections and live in a tent and get to know these plants in a really sort of special way. Like I've spent a lot of my life around them and looking at them and learning about like who they are, how they interact with their environment, and vice versa. And one of those trips in 2014, the inset photo is my family, my wife, Rachel, and my kids, Izzy and Jackson. That trip, 6 weeks living out of a truck in a few tents is like probably my life highlight. And it was awesome, right? But having like lived around these plants and gotten to know them and also grown them here at Bucknell for the last 8 years, like about 20 different taxa here in our greenhouses, it made me realize that what we really need to understand these things is a sort of a truly comprehensive phylogeny, right? And we've as you'll see in a minute, we've made attempts. We've generated phylogenies over the last 10 years, but we still have never really made one that I felt super confident in, and especially if I'm thinking about how these sexual systems have evolved, right? And who's sort of transitioned from who and what order did they come in, because I'm just really curious about knowing that. They're really interesting to me. And so it was really Angela McDonald, who was a postdoc in the lab 2 years ago, who said, Chris, if you're going to do this, you really need to do it right and make it sort of the final phylogeny. And we always joked that it was PHY for final phylogeny, which is funny. I'm imagining a couple of you chuckling at least a little bit. It's not that funny. It's more of a dad joke kind of thing. But Angela said, we're going to do this. And she said it while eating an apple off a knife in the outback. And that's when I knew she was really serious. So what I want to present to you for the next few minutes are some slides that Angela put together as part of a presentation we did at Botany 2020, talking about our attempt to now sort of do this final comprehensive phylogeny for these 40 or so species that represent 3 different sexual systems in Northern Australia. And, you know, as I get into this, I just want to say that it's Angela, it's myself, but it's also a lot of other people that have contributed over the years to this, including collaborators in Australia, traditional landowners who allowed us access to plants and land, herbaria that shared with us specimens, and all sorts of other folks, right? This is a long-term project that's taken a lot of effort and has meant that we need to work with a lot of different people. And as we get into this, I want to remind you that these are just amazing, beautiful critters, right? They're loaded with these amazing prickles. They live in amazing places. And so getting to know these and now then trying to uncover their evolutionary history has been, you know, a bit of a dream. Right? So we've tried this before, this sort of getting a phylogeny for these taxa. In fact, my PhD dissertation at University of Connecticut had 2 chapters in it that were phylogenetically based. The one chapter was based only on ITS, and that was published in Systematic Botany. And then this is the second one, a paper published in 2009, that is really based on only 2 loci, ITS and Turn K Mat K. And what did we learn from this paper, right? Well, we found that we basically have 2 distinct clades of dioecious taxa, one that occurs in the Kimberley Plateau and another that consists of just a few species that are part of that Kakadu National Park region that I mentioned as being in the top end. So that's interesting. So it seems like maybe we might have, you know, given their placement in this tree, Dioecious may have been evolved just one time, and then we have this sort of like radiation of that lineage. We also found really decent support for that the Andromonisi group actually also represents a monophyletic clade. So that's cool. That's 2009. That's 2 loci, but we really don't have a lot of backbone support allowing us to determine which way any of these unusual reproductive systems have shown up in the evolutionary history of these plants. 10 years later, and this is just last year, after lots of years of sort of working on this data set using marker miner and a few other protocols, we published a 7 loci tree. And what did we find there? Well, we found essentially great support again for these 2 clades of dioecious taxa, one in the Kimberley, one in Kakadu, but the potential that this might have actually evolved 2 independent times, right? And also really great support for a monophyletic group among those Andromonisi taxa, but very poor support again in the backbone, and thus equivocal support for any kind of hypothesis for how these breeding systems have evolved. I just want to point out that this paper that came out last year featured 4 undergraduate authors, including your very own Emma Frawley, who just did the introduction to this talk. Right. We're not the only ones who do Selenium phylogeny. That's a huge group. People all over the world are working on it. So just to show you one example of that includes some of our taxa. Here's a paper came out in 2013, Sarkinen et al. And they use 7 loci, and here's what they came up with, right? Dioecious evolving twice, 2 monophyletic groups, one in the Kimberley, one in Kakadu. And then again, good support for a monophyletic group of Andromonisi taxa. So these clades keep coming up, and they're pretty well supported. But again, we don't know essentially where the breeding systems are coming from, what they're transitioning from, right? That question. So what was our goal here, right? Well, actually finally have a tree where we can resolve a topology that allows us to sort of look at the conflicts across sort of like different gene regions, but also that we look at across different papers that have published this, and really with a focus on looking at how sexual systems have evolved. So we sampled from all over the world, but of course largely in Australia. A lot of this were wild collected, some were collected in cultivation, and about 85 of these were also collected from herbarium tissues. 1 to 5 accessions per taxon, and we sort of oversampled to some degree with the Australian things, particularly taxa that are pretty variable, right? But the most important thing here is that we were sort of working on a new platform. Arbor Biosciences released a set of universal probes in the last year or so called Angiosperms 353, and we're one of like a couple handfuls of groups that in this past year have attempted to apply these universal probes to our particular study system. It's an amazing concept, right? This idea that you could basically have the same 353 gene regions across any lineage in the angiosperms, right? And that any of us can tap into that, not have to develop our own probe sets, save that time, save that money, important for a guy like me at a PUI. And then if it works, right, you've got these really cool data sets that can also be compared across each other, which is super neat. So we gave it a whirl, right? Excuse me. I can't stress enough how important herbarium collections were for this. This is New York Botanic Gardens herbarium. There's Angela and former master student Cheyenne Moore, and there's my mother, Barbara in the background, who was also a big help on that day. She also made me this quilted wall hanging behind my shoulder of some Australian Selenium. She's a nice lady. These are what we did. So this is like the pipeline, and I'm not going to go through this, right? But just for those of you who are really interested in this thing, this is what we did. And if you've got like legit questions about how this stuff works, that Angela is the exact person for you to reach out to and talk to. So that's what we did. And I will show you what we got out of it, which is just a whole crap ton of data. 5.25 million reads, right? And each one of those being on average 755 base pairs long, which is ridiculous, right? My first chapter of my PhD that got published in a good journal was just ITS, and that was about 750 base pairs long, and that was it, right? And this is the kind of data we're able to generate. I will never not be impressed with this amount of data. It's ridiculous, right? And so when I saw all of this, I thought, well, of course, we are going to figure this out, right? We're throwing all of this data at this phylogeny. We are going to know exactly how these breeding systems have evolved, right? So let me show you some results. So this is the angiosperms 353 results. This recovers pretty nicely about 9 different lineages. We included a ton of outgroup things, right? Really to cover ourselves, to make sure that we're absolutely certain that our groups that we're dealing with are really sort of all monophyletic, and that, we wanted to make sure we really covered Australia, and largely a lot of the old world spiny Seleniums are in this mix too. And so I'm not going to spend a lot of time talking about these outgroups, which in themselves tell a lot of interesting stories. But what do we get with some of these clades that were recovered in the other papers, right? So there we go. There's our monophyletic Andromonisi group, right? That's pretty cool. There's our first clade of Dioecious taxa, the group from the Kimberley Plateau. And there's our Kakadu Dioecious taxa, right? So very a tight association between those two dioecious groups, perhaps supporting a single origin of Diaece. But here's the thing that's really interesting. The one group I haven't shown you on any of these other trees is this group of about a dozen hermaphrodite taxa that co-occur with these Andromonisious and Dioecious species. And it's largely because previous analyses haven't really shown a lot of support for them being too closely allied with what we considered previously to be our in-group. But here's what we find, right? We've actually got a decently supported, actually a really well-supported clade of all of our Dioecious taxa and all of those hermaphrodite taxa. And this is, given the sampling, this is kind of the best evidence that these sets of species are actually pretty closely related. So we've got a clade consisting of dioecious species and hermaphrodite species. Interesting. Angela also happened to recover 40 roughly complete plastomes, right? Just amazing that you can even say that. And they show very similar things, right? So there's our outgroup stuff, but very strongly supported Andromonisious group, strongly supported clade of Dioecious taxa, and then there's our hermaphrodite group, which isn't quite the same result we see in the previous tree, but we're going to roll with it, right? So all sorts of important stuff comes out of this for the outgroup taxa, but I'm not going to really say much about this, except to say that what we're seeing is some really interesting broad trends related to the species that we were looking at, and that is that we see multiple lineages in which we have earlier divergent lineages that are sort of wet tropical in rainforests, on the edges of rainforests. And then within those clades, we see these transitions to sort of more arid, semi-arid, more drier conditions, which is interesting, right? We also see that within those lineages, the taxa become more and more adapted to particular high levels of disturbance, most notably to fire. And that's important because we see this broadly, and other people are seeing this across the nightshade family, the Solanaceae. But we also see this within our in-group. Within just the species that we're looking at, we see the same trend, right? We see this trend that we basically have these sort of earlier diverging lineages that seem to be sort of more wet tropical, that have transitioned to being adapted to semi-arid and arid conditions. And actually, that's not that surprising, because that's the trend we've seen in terms of the climate in Australia in the last 3 to 4 million years, and our lineage is sort of mirroring that change, which is pretty cool, right? And also mirroring that sort of change to being more and more disturbance adapted, particularly to fire, right? And that's pretty fun. That's pretty cool, right? But before I tell you what we see with sexual systems, I just want to say a little bit about how there's actually some nodes here where we actually don't have a ton of support. All my dreams about having like more than 300 gene regions, this massive data set of more than 5 million reads. There's no way we're going to have poor resolution anywhere. And it turns out we do. There's 2 here that I'm not going to say much about, but here's this one right there. in the exact clade where we want to know how sexual systems have evolved, we've got a poorly resolved node. How can this be possible, right? It just is, right? So how it's possible is that we have all sorts of gene tree discordance. And I'm not gonna spend a lot of time on this, but what I want you to see is that at every node, there's a bar, right? And the more red it is, that's the more gene trees that actually reject that topology at that node, right? So I'm just gonna do this. I'll just fill them all in, right? There's a lot of red, right? And including a lot of red right here at this critical node, right? And it turns out that these three clades that are supported, we just don't have a ton of information about how they're related to one another, and thus how the sexual systems have evolved, right? That important node is actually only informed by very few genes, right? Any of these three competing topologies that tell us how these breeding systems are related to one another is actually, you know, just as likely, and based on only a small number of genes relative to the many, many that are in our data set. So we think this is related to this being just a group that is recently radiated, right? So we've got all sorts of reasons to believe that we have incomplete lineage sorting and that this is all just happening, you know, right now in real time in Australia, right? So maybe this is real, right? Maybe these polytomies that I've been getting for this group since 2006 with just ITS that we're still getting now with 5.25 million reads is really just reflective of the fact that this is a real deal, right? This is as good as it's probably going to get. And I've been uncomfortable with polytomies, like maybe some people, for a long time, and I'm actually starting to become pretty comfortable with them, right? There's a little mystery there, and maybe some of that mystery is fun. Now, we also, given all the people that are working on Selenium, have actually quite a bit of evidence that these polytomies are just consistently going to be all we get for a lot of the genus Selenium. There's a big project looking at the entire genus led by Edeline Gagnon, and a number of us have contributed data to this. That'll be out soon, basically saying like all over the phylogeny for this genus, we have evidence that essentially everybody's recently radiated, right? We have ongoing evolution. In Australia alone, right? This shift to a more arid continent is not done, it's ongoing. And we're seeing species sort of evolving and radiating essentially, you know, right under our noses, which is exciting and means we may never have a fully resolved topology. The other thing that has complicated things is that we know that hybridization has also been happening, right? So we've got taxa that have diverged and maybe come back into secondary contact. And we have evidence based on some of the analyses that Angela just recently ran that we have at least 3 or 4 clades within our group where we see lots of evidence for recent hybridization. And of course, that muddies the waters, right? But what is our most likely topology say about sexual system evolution? This is why you bought your ticket today, right? You went down to Ticketron at the mall and put down your cash money and you bought these tickets, right? That's what you would have done in the 1980s, but this is 2020, so you didn't do that. But here's what we got, right? Here's what we got. So this is, I love this tree. I love this tree. What this shows us is sort of the likelihood at any given node of the condition being one of those 3 breeding systems, right? So where purple is hermaphroditic, yellow is dioecious, and green is andromonisious. And so this is on a backbone of sort of an understanding that the ancestral condition coming into these old world species is probably hermaphroditism. And I want you to notice that already sort of at the base of this tree, what we see is a big mix of hermaphroditism and Andromonisi. There's a lot of toggling back and forth, a lot of lability being shown among those pretty closely related taxa. And that's pretty interesting to begin with. But yet, our groups, here's our Andromonisious bush tomato group, right? Monophyletic, everybody's Andromonisious. Here are our 2 clades of dioecious taxa from Kakadu and the Kimberley. Everybody in those groups is dioecious. And here we have this clade that we now associate with these dioecious taxa really for the first time in a fully confident way that are all hermaphrodite, right? And what you can see here is we've got a couple possibilities. We've got Andromonisi here, likely at this node, but the question is, do we see a transition from Andromonisi to hermaphroditism? Do we see a transition from Andromonisi to Dioecious as proposed by Anderson and Simon? And even here, do we potentially see an actual transition from Dioecious arising first and then transitioning to hermaphroditism, which seems like it totally shouldn't be the case. All the literature on Dioecious that has been written for decades has said that Dioecious is basically an evolutionary dead end. You don't come back from choosing unisexuality. It shouldn't be possible that you can actually somehow go from being just male or just female to now being bisexual again. But the truth is we have evidence for both the possibility of Andromonisious things becoming fully hermaphrodite and Dioecious things becoming fully hermaphrodite. And it's all about having actually looked at real plants. So could we go from Andromonisi, where you've got a basal hermaphrodite and a bunch of males, to having a fully bisexual inflorescence, a fully bisexual plant, right? And the answer is yes, right? Based on even just what we are seeing on real plants in our greenhouse. We know that there's such a thing as strong Andromonisi, which means you've got just the bisexual flower at the base and a bunch of males, right? That full separation. But there's also something we have, tend to call weak Andromonisi. And that is this sort of proclivity for a lot of these Andromonisious taxa to sort of switch some of these males in the upper part of the inflorescence over to being fully bisexual. So we see a lot of variability in style length in these flowers. Sometimes the more distal flowers stay male, and the ones that are, closer to the basal hermaphrodite begin to switch over, right? And this basically is a way of transitioning an inflorescence from being strongly Andromonisious to being fully hermaphroditic. Like you saw in that tree, there's a lot of lability between those two breeding systems, and we're seeing evidence for it in our plants that we grow here in the greenhouse. So that's possible, right? But is it possible to go from a dioecious system to a hermaphroditic one, right? This shouldn't be possible, given what we see, what we might imagine. But what we actually see in the greenhouse is that this is absolutely possible because of the preponderance of something called leaky dioecious among the Australian Seleniums that we have in cultivation. The way this looks is that your females that are, remember them, morphologically bisexual, but making these non-functional pollen grains, nothing really happens to them. They just sort of keep doing what they're doing. But what we see in our population is that a lot of our male plants, right, among a whole population, you will start to see individual plants that actually start to leak. That is, male flowers are beginning to express themselves as bisexual. Sort of the dirty little secret here is that every male flower in the genus Selenium has a super tiny little vestigial gynoecium that is non-functional. But in our taxa, every once in a while, that gynoecium actually fully develops. And you see this sort of less robust, sort of somewhat elongated style sort of peek its way out of the anthercone. And those are functionally hermaphrodite. You can pollinate those and get fruit set. So we have evidence among our species in cultivation that males actually can switch to being hermaphrodite. And so this reversal, right, within these dioecious taxa to a hermaphroditic condition is actually not that surprising based on what we've seen. Some might even say we didn't even need the phylogeny to sort of understand that you could have this transition occurring, because the answer is right there in the flowers that we're seeing in the greenhouse. And yes, those flowers will sometimes set a fruit, as you see right here, OMG, a fruit actually forming on a male plant. Now this is interesting, right? You could think that, well, this is just an aberration in your greenhouse. It only matters if you see it in the field. So we've been looking, right? And so here's a species, Selenium asymmetrophyllum. This is a sort of normal male inflorescence here, right? And here is one where we have the flower at the base of that male inflorescence that has leaked and actually formed a fruit. Pretty exciting. Here's another species, right? Selenium dioicum, widespread across the Kimberley region. And I've encountered actually now all the time with males that are clearly mostly all male, but have these occasional little small diminutive fruits formed at the base of the male inflorescence, right? Okay, so that's cool. Yes, they make fruits. Your next question is, oh yeah, but do they make seeds? And can those seeds grow into baby plants? And they can. And the reason we know that is because we had two awesome undergrad students who did all sorts of crosses with all sorts of flower types, Ian and Dan. And Dan individually, as part of his honors thesis, did more than 4,000 individual hand pollinations. He actually was trying to see if you could hybridize all the species in our study group with one another. It turns out everyone can set fruit. Everybody can hybridize with everybody else. More support for the fact that these are recently diverged groups and helps me a little bit better with those polytomies. But my point here is that every time a male plant in our greenhouses has leaked, we've pollinated that flower, right? So these are male plants that occasionally express as bisexual, and we put pollen on that stigma and see what happens. The first species that we ever had success with is a species called Selenium subjunctum. This is the first fruit we ever made with a leaky male and a dioecious species. Very exciting moment, right? Up here in the right-hand corner, which might be partly obscured by my head, this is what a normal Selenium subjunctum fruit looks like. And this is what a leaky fruit looks like, right? Much less developed. The calyx is not fully enclosing that fruit, and they are like a quarter of the size. Whereas a normal fruit might have up to 400 seeds, these might have 10 or 40 seeds, right? So much smaller fruits, much fewer seeds, but they're fruits being formed on male plants. And what's really cool about this is that my advisor at UConn, Greg, at the same time that we were doing this, also had a species called Selenium polygamum that's from the Caribbean that is dioecious, and his male plant also leaked. And so he pollinated that leaky flower, made a fruit, and made seeds. So here we were, each of our labs with seeds made on male plants, and we planted them. And what happened was amazing. Right? What happened was amazing. In both species, here's what we found, right? And let me just say that every time I've encountered a population of dioecious seleniums in Australia, across 6 trips, I always go in and count the number of males and females. And it's nearly always 1-to-1 male-female. When we germinate seeds here at Bucknell, it's almost always 50% male-female. Once they go to flower, we pretty much have half and half. If you take the seeds out of a fruit made on a male plant, that's not what you get. What you get is 100% male recruitment. Every single seed produced by a male plant just grows into more males. And you know what they can do? They leak, right? So now you have a population that's 100% leaky male, right? Which is really 100% hermaphrodite, okay? Now, Greg and I both tried it with a second generation. We allowed that first generation to grow. They leaked. We pollinated those flowers. And the F2s, 100% male, right? Everybody is a male and everybody can leak. And now you've transitioned to a hermaphrodite population, right? Yes, you have to account for what happens to those old females, right? But they're being replaced at a much higher rate now that, you know, one part of your population makes nothing but males. And there you have it. Like, it's actually possible to transition from what many people think of as a sort of reproductive dead end, an evolutionary dead end. You become dioecious, you stay that way, to this possibility that you could actually evolve an entirely new lineage of hermaphrodite taxa from your dioecious things. That's awesome, right? At least I think it's pretty awesome. And I think it makes a pretty good story, right? And so I'm going to transition here for the last bit of my talk, because I want to talk about good stories, right? So, you know, some of you might be like, okay, this is pretty awesome. Like, you know, cool reproductive evolution stuff going on in seleniums. I actually thought I'd talk a little bit more about like fruits and seed dispersal. If you ever want to know more about that, it ties into this story really nicely as well. But those kinds of stories are great, right? For us, you know, as scientists, those are interesting. We'll get these things published, right? But I'm not sure they'd carry a ton of weight sort of as a media story with the public, right? Not everything we do, it gets us that way. But what I want to do is share with you some ideas I have about how we can actually parlay a lot of our scientific outcomes into stories that the media will pick up as a way to promote the sciences, right? And as I'm saying this, I'm going to guess that there are some of you with this little bit of your brain that's like, oh yeah, that's not what I do, right? I'm a scientist. You shouldn't be asking me to go out and do science communications and speak to the public. It's not my thing, right? And maybe some of that is a resistance to sort of looking like a self-promoter, right? But what I would say is that we are at a time now more than ever. And if I had given this talk 10 years ago, I would have said now more than ever. It's always now more than ever, right? But it really is, right? At a time where all sorts of people have a fundamental distrust in the sciences, right? So what better time than now to actually begin to embrace this notion that, you know, what we might feel uncomfortable about in terms of self-promotion is actually promoting science itself by talking about our scientific outcomes and trying to find ways to sort of share those with the public. We might actually be sort of saving the scientific endeavor itself, right? So what I want to tell you about are a couple of really intentional ways that in the past few years, we've made an attempt to develop media relations strategies to promote some of our scientific findings. And the place where we've been doing that is with our new species descriptions, right? And there's a reason. for that, right? So what I'll do is I'll first say that the first species that really took off for us is this one called Selenium watneyi. We had this thing found. It was actually collected with my family on that trip in 2014, which is pretty exciting. And we had it described. We had the paper basically written, the whole manuscript, but we didn't have a name, right? And I came in one day to the lab group and said, hey, here's an idea. You may have heard there's this new movie coming out based on the book The Martian by Andy Weir. Matt Damon is going to play a space hero botanist, which is just a ridiculous opportunity, it seems, right? So we decided to name this species after his character, Mark Watney, the NASA astronaut space botanist. And, you know, before you sort of poo-poo that, it's totally legit because the species he lives off of on Mars is potato, and that's in the genus Selenium. So this is like, it's all good, right? Totally rigorous choice, right? So what do we do here? We wrote about it. I put a thing out in the HuffPost, said, we're going to name this species after this. I wanted to tie it to the release of the movie. And then we all took the plant to the movies. We had plants in cultivation. We took it to opening weekend. That generated some local buzz. It was a lot of fun. We thought, okay, that's pretty cool. And then the story just got picked up all over the place, right? Including places like Time Magazine online and like hundreds, literally hundreds of outlets all over the place. And there's undergrad co-author Emma Frawley, your own Emma Frawley right there, right? And so we got all sorts of really good buzz for this species. And it was really awesome to sort of see people engaging with our science this way and also seeing, you know, our student co-author get attention for this as well. Did it really matter? Did we really make an impact? Yes, because the species is mentioned on Wikipedia. All right, so the next species that we worked with was this one that we eventually named Selenium ossicruentum. Again, Emma is a co-author on this paper as well. And we decided to take a different angle on this one. For this one, we went to the local middle school and we engaged 150 7th grade life science students. And we sort of gave them all the information they would need about this species and then said, okay, everyone write an essay where you come up with a name, Latinize it, do the whole deal, and then propose it to us as a scientific group, and we will pick one of your names. And we ended up doing that. This name, Selenium ossicruentum, is derived directly from actually a couple different students who wanted us to focus on the morphology, the morphology of the fruits. When you cut open unripened fruits, they look like they are bleeding. So there's the cruentum part. And when they ripen fully, they're hard as bones. So that's the ossy part. So we have this thing that basically took on the name as sort of the bloody tomato or the blood bone tomato. And between the 7th graders and that cool thing, that also got us in the news globally, all over the place, including this really awesome article on Science Friday, and eventually got us onto the top 10 new species list for 2017, which was really awesome because that list is mostly always animals, right? And so to get a plant on there was really fun. Right. So great. Another species where we got some buzz. Is this sort of like empty sort of bubblegum buzz, or does it also translate to some attention for the actual science, the primary literature? And the answer is yes. All right. So this is, you know, a paper that has now, as of today, been viewed more than 11,000 times at the journal Fidokeys, open access journal, where we published this, which is outrageous, right? How many people would actually go and read this paper if it was just sort of a standalone new species paper from Australia? This is all about us and a willingness to sort of, you know, step out and promote the work that we're doing. There was a time where you could find, there's the Google search, 6,500 different places online where this name was mentioned, right? And it also has translated into sort of this cool altmetric score, right? So this is, if you know, a lot of journals now are doing this. This is sort of a measure of sort of the buzz created across all sorts of platforms about your paper. And 162 is a pretty decent altmetric score, right? Well, what I want to tell you about is our last species that blew that altmetric out of the water, and that's this one here. So this is a species that was collected in 1974 by a great botanist named Peter Latz. And this is the annotation label from that collection by David Simon, who wrote the monograph. Here's what Simon says. That makes me laugh every time. Here's what David Simon had to say. He said, "This has me puzzled. It is obviously one of the Andromonisi species, but lacks the male racemes above the fertile flower. I have not seen its like before, but I think it's some variant or aberrant." And you know where that aberrant went? It went in the indet folder and got put in a cabinet. And for 40 or 50 years, nobody really dealt with it, right? And so we came around in 2018 and again in 2019 and actually went out and collected this thing out in the wild. And we found the same thing. That frustration about like not understanding what sexual system to put this into. You've got really 3 options in this region, and it didn't seem to sort of fit neatly into any of them, right? Enough to make somebody say, you know, I'm not even going to deal with this right now. That's the part that we found most exciting, right? And that's why we named it Selenium plastisexum, to sort of recognize the fluidity of the sexual condition in this. And given how much we're into this kind of stuff, that ended up being really a perfect way to name this. And it's sort of a normal taxonomic paper describing a new species. It's got Heather on it, who is a senior right now, as a co-author. It's a nice paper, right? But we ended the paper with this sentence: the notion of a constant sexual binary consisting of two distinct and disconnected forms is fundamentally a fallacy. Now we're saying in Selenium, you know, the binary thing is bunk. Maybe we're saying across all organisms, this binary thing is bunk. Maybe we're saying in people, same idea, right? And then we wrote a press release that sort of talked about that idea a little bit more, and that generated really cool buzz. And that ended up getting us in the New York Times, right? Which has a certain way of you know, increasing the buzz you get about your science. This paper is relatively recent, and it has just gone over 9,000 views, and our altmetric score is ridiculous. That, I don't know how well you know these scores, but I can't even believe that number, right? The biodiversity papers, taxonomy papers in particular, don't tend to do that. And it's all because for this particular paper, we decided to take a little bit of a stance, like make a statement, right? Which in the paper is sort of subtle, right? But it became a message that a lot of people grasped onto, and we were pretty pleased with the way it went. But what I'm most especially pleased about is that it ended up amounting to this really incredible collaboration with an artist named Erin Johnson. That New York Times article, she reads in the newspaper and gets in touch with us and says, you know, I'm intrigued about this species, right? I'm an artist, not a scientist, but I love this idea that you're sort of promoting with the naming of this species. And she came and interviewed us. She shot video, she shot photographs. We got to sort of sing on camera and do all sorts of things a little bit outside of some of our comfort zones. And all of that stuff that was generated from that interaction became the core of what have now been 3 art exhibits at 3 different museums. First at a museum in an art gallery, in Los Angeles, and then at the Center for Maine Contemporary Art. And currently, an aspect of this is showing. It's a video called "There Are Things in This World That Are Yet to Be Named," which is a quote from the Times article, where you can see Tanisha and Cheyenne Moore and Ariel Antoine doing work with these plants and actually singing some songs about seleniums, which you don't get very often. So I encourage you to go check that out. This has probably been like the coolest outcome that's come from us being willing to sort of talk about our work and promote it in ways that maybe most of us as scientists don't get trained to do or are not very comfortable to do. But what I want to tell you about is one last thing that is a really good example of the type of things that can happen when you put your stuff out there. And this is a new collaboration that we're developing with some colleagues at Penn State, Rebecca, Doug, Chloe, Amy, our collaborator at Charles Sturt University, Dale Nimmo. And you see Tanisha as part of this project, as is current senior Jeff Hine. And this is a project looking at just one of the species that we've been studying and that we have in cultivation here, called Selenium diversiflorum. You see, that's the native range, the purple dots. And there's a lot of it within the Martu native title. This is sort of land where the Martu people reside. These are folks that were forced off their land for a good part of the 20th century and 30 years ago returned to their land and started to sort of readopt traditional cultures, including stuff that surrounds the use of certain plants. And it turns out they use this species, Selenium diversiflorum, which they call wamala, in a pretty serious way. And it has been used, you know, for generations, likely for thousands of years. And so this is a project where we are partnering with cultural anthropologists at Penn State in a way that I never would have imagined doing before. And the only reason it happened is that Chloe, who's a current PhD student, was Googling the name Selenium diversiflorum and found out there was only one other person in the entire country who's ever tweeted about Selenium diversiflorum, and it was me. Right? So she saw our pictures from the greenhouse and got in touch with me and said, I can't believe you're only 50 miles away. And I can't believe that you also study this plant. And I also can't believe you actually have it growing. We were the only place in the world outside of Australia where you could see this species. And that started a collaboration that actually resulted in a successfully funded NSF grant in the last year that I'm super happy about. So what are we doing? We're coupling the work that they've done, right, in all sorts of areas, including sort of ethno-historic observations. What you see there are people in the current day gathering up wamala, Selenium diversiflorum. And what's really interesting about the way this has been done forever is actually what you see here in this upper right-hand photo. So these are folks processing these fruits. The fruit wall is apparently delicious, but the seeds are something you do not want to ingest. They're super bitter. They're not pleasant to eat. So in order to process these fruits, you cut them open and you scoop out the seeds and toss them to the side. Now here's what's pretty cool about this is that historically, these have been fruits that have been sort of eaten while out bush, right? While out traveling in the desert, largely in these arid conditions. And what you see here largely are places where this species occurs along historical pathways that people have been traveling for millennia, perhaps, right? And the way we know this is that each one of those dots also represents a camp area. And so these are spots where there are water sources. So these are great places to stop for the night. You've got water and you set up a fire, you process your wamala fruits, you dump out your seeds, right? You see where I'm going here. And so now what we have are these sort of casual gardens of this species that exist at every one of those points on the map. This vast landscape where we have these gardens of Selenium diversiflorum that have been influenced by the movement of humans and maybe the selection of humans for perhaps centuries. So what we're going to do now, and this is where Jeff and Tanisha have been doing a lot of work, you see them in that picture for the last few weeks, is we are now seeing if there's a genetic signature related to this. If we compare the populations that are around these camp areas and along these paths to wild populations that are off of these paths in this really vast landscape, do we see a structure that is related to the use of this plant by people? And like, we're crazy excited about this. It's not something I ever imagined that I would do. And I owe it in a lot of ways to Twitter, right? So that's really all I'm going to say about Selenium. I do want to say that all of this really just amounts to, the fact that plants are cool too. And I hope you do watch the video series. It's really fun to make, and I hope you check it out. And I have other stuff I can talk about, but for now I'm going to end right there and say thank you for listening.
Thanks so much, Doc. That was a fabulous presentation. I think you packed in a lot of neat stories in there. And I loved kind of rehearing stuff from my undergraduate career. This was fun for me. So it looks like we just have a couple minutes, and I'm going to pull some of the questions from the YouTube stream just to get a quick response there. Someone asked, a professor at WashU said her 5-year-old is asking all of the questions and kindergarten has not covered polytomies yet. But it's too bad.
Flowers? We're talking about flowers.
The first question is, we really need ancestral state reconstruction methods that can deal with polytomies. Any insights about this?
No, but I do think you're right. And I think the fact that so many, the amount of data we're generating and phylogenies that are showing what pretty well supported topologies with all this gene conflict and lots of polytomies will now necessitate that we come up with tools to do this. So I don't have an answer for that, but I know it's sort of critical, and I hope that we can use some of them.
Okay, another professor or another person on the chat asked, are there developmental studies about the two types of pollens? And when referring to inaperturate pollens, you said the openings are being closed up. This person is wondering developmentally, are they being closed up or are the apertures not being made initially? It seems that switching between two types of pollens might be some simple genetic programs. Do you have any thoughts about this?
Yeah, so on the closure, we've done a little bit of work. What we find is that that's a really sort of thin layer of, I guess, exon, right? It's almost like spackling is how we sort of compare it. It's very thin. It actually, we're not quite sure if it sort of is a late deposit or if it's just this little bit of that layer that essentially just doesn't fall away, right? But we actually do know quite a bit about those pollen grains. In fact, I can show you this right here. Like, we've actually done some comparisons between those two pollen grains and looking at sort of like their nutritive value. They basically develop exactly the same. The male gametophyte essentially is the same inside of these things. You just can't get out if you're in the inaperturate grain. And we've actually shown, I'm just going to show this because they're here, is that we do see some difference in nutritive quality. So they've got kind of everything that these pollen grains basically have everything in either thing. They just, one of them can't can't germinate, right? And so it's pretty interesting, right? And these levels of protein that you're seeing here are actually really high relative to most types of pollen, simply because of the system that this is. But we're currently trying to figure out why male pollen actually seems to have a bit more protein than female pollen. But that's something we're working on now. Yeah.
I think I will maybe leave you with a comment from Sandy Knapp, who says that, hey, Chris, I think this gender fluidity is super common across all of Selenium. So maybe Simon's Andromonisi is really just leaky Dioecious.
Yeah, it may be. Sandy lurking in the background there. You probably have all sorts of stuff that we can talk about right now. Thank you for being there. Right. Can I just say one last thing real quick? All right. I just want to sort of make the point, those of you who are postdocs and grad students thinking about a life in academia, this is all work that has happened at an undergraduate institution that includes the training of undergraduates. So if there's one thing I want you to get out of this, is that it's absolutely possible to be both a teacher and a scholar at institutions like this. And if we look at these numbers, 4,000 schools in the US and only about 200 are considered R1 institutions. Everywhere else, the focus is on undergraduate graduate education. So if you're gonna get a job, it's more than likely gonna be at a school like this, right? And it's possible to do really good work and train a bunch of cool kids, really, like Emma.
All right, thanks so much everybody who came and left comments. And if there is anything else you want to ask Chris, there's a reception happening after this. And so I hope to see you there. Thanks so much, everybody.
Thanks, everybody.